In this video, we're going to take a brief look at indicators and some common indicators that we might come across. So, to start with, how do we know that a solution has been neutralized? How do we know whether it's an acid or a base or whether it's neutral? Well, for most neutralization reactions, there's no way of knowing the solution has been neutralized because there's no actual visible sign. Most of the time, the salt that's created is still soluble in solution, and so it doesn't look any different. So we need something to be able to tell us that a neutralization has occurred. And that's where indicators come in. So we can use pH indicators to determine whether a solution has been neutralized. So there are some common ones, and many of them are in the IB data booklet as well. But let's just take a look at a few of the popular pH indicators that we come across quite regularly in our lab. So the first one, phenolphthalein, it's quite a mouthful to say. Um, that has a clear color. It's actually colorless in an acid, but it turns pink in a base. Bromothymol blue is yellow in an acid and it's blue in a base. Litmus paper. You probably came across this one in grade 10 science, but this one is red in an acid and it's blue in a base. And then methyl orange is another popular one. That one is red in acid and it has a yellow orangish kind of color in a base. So, like I said, these are just a few of the popular indicators, but there are many more. So, you'll find charts like this everywhere in terms of what sort of indicators are available. And you can also sort of see where those color changes happen. So, there are indicators that work well at lower pH ranges. There are indicators that work really well at sort of neutral pH ranges. And then there are ones that work quite well at basic ends of the pH scale. And the way you sort of read this is you can take a look, for example, phenol red is a yellow color in more acidic solutions, and then its sort of changing point is around pH 7, 7.5, and it's going to then change into a red color. So it would be yellow in an acid, it would be red in a base, and so on. So you can use scales like this to determine the colors that you might expect with different indicators. Now, one more final thing. So we can use that pH indicator to tell us when we have hit a neutralization point. And the reason we can do this is because when you have a neutralization reaction, the moles of the acid are going to be equal to the moles of the base. If you remember from previous videos, that is when we're going to hit that neutralization. And when we hit that neutralization point, that's when you usually see that color change of the solution from the indicator itself. So when you get that change from going from clear to pink with phenolphthalein or yellow to blue with bromothymol blue, that tells you that you have hit your neutralization point. And this will become more important as we get into some of our other reactions and calculations a little bit later in this course. So that's it. That's a general overview of indicators for now. Let's move on to our next task.